Hello, my name is Alyssa Lopez, and I was assigned protein KRAS, mutation glycine amino acid number 12. It's replaced with cysteine G12C. The KRAS gene is what makes the KRAS protein. This protein relays signals from outside the cell to the cell's nucleus. The signals instruct the cell to grow and divide, which means proliferate, or to mature and take on specialized functions, which means differentiate. The KRAS protein is a GTPase, which means it converts a molecule called GTP into another molecule called GDP. In this way, the KRAS protein acts like a switch that turns on and off by the GTP and GDP molecules. To transmit signals, it must be turned on by attaching to a molecule of GTP. The KRAS protein is turned off when it converts the GTP to GDP. When bound to GDP, it does not relay signals to the cell nucleus. I would categorize the type of function that the protein holds as a protein that relays signals. This is because the protein communicates with the nucleus to send signals to it to then proliferate and differentiate. This protein is normally tethered to the cell membrane. The gene is on the short end of chromosome 12. It is found in mammalian cells. The protein becomes more active because when mutated, the protein has the potential to cause the cell to become cancerous and plays a role in some of the most common and deadly carcinomas. When mutation occurs, they amplify the signal pathways that lead to forming cancerous cells. This leads to my 3D model. This is the 3D model of the cell with the KRAS protein. This is the KRAS protein. We have the nucleus right here. And what happens is it goes from outside of the cell. This is the RTK. It goes from outside of the cell, then goes inside, and then how this is tethered to the cell membrane, and then it sends signals to the nucleus to then proliferate, make the cell divide, proliferate, and differentiate. This is the normal. Now, when it is mutated, this is what happens. So, they send stronger signals to the nucleus, and then the cell starts to divide uncontrollably, causing the cell to become cancerous, and then this sends to the rest of the body and causes organs to become cancerous. And this is what I have. And this is where the mutation occurs, right here. The KRAS protein, that's where the mutation occurs. So the main functional groups, there are two functional groups, are carboxyl group, which is COOH, and an amine group, which is NH2. This is the simplest amino acid. Also the simplest side chain. It's composed of a single hydrogen atom. That's the side chain. So these are the properties of glycine side chain. It is hydrophobic because it's nonpolar. It's the smallest of the amino acids. The type of bonds glycine can participate in is bonding with other hydrophobic side chains. It also repels water. So the properties of the new mutated amino acid side chain is 3 ionizable functional groups. It's including carboxylic acid, an amino, and a sulfhydryl group. The side chain is thiol, which is SH, and slightly polar, but its polarity is so mild that it's unable to properly interact with water, making it hydrophobic. Cysteine side chain is capable of forming a disulfide bridge, which is covalent bond between two sulfur atoms through side chain oxidation and removal of two hydrogen atoms, hydrophobic, so it can form with other hydrophobic amino acids. G12C, which is the mutation that I did. This is mutation G12C, which is usually found in lungs, for lung cancer. It's usually lung cancer. It is a single point mutation with glycine to cysteine substitution at codon 12. When mutated or changed, it can cause normal cells to become cancerous cells. The cells multiply out of control as well. So when they multiply out of control, they become cancerous. The KRAS gene belongs to a class of genes known as oncogenes. A mutated KRAS protein allows cells to grow and spread in the body. Also, the self-destruction of cells called apoptosis. KRAS mutations can not only affect the gene itself and the expression of the corresponding problem, but can also influence the expression of other downstream genes involved in crucial pathways regulating cell growth, differentiation, and apoptosis. So I would say that the limitations are on the 3D model is they're not able to show the active and inactive states for the protein. It's not able to show cell division, and it's not able to show how the cell travels throughout the body and causes cancer throughout the body. Thank you.